I had a friend who flipped his sleep cycle and just come in at like, 11 at night and work until 7 in the morning. And like, that was fine. No one really cared, right?
Hi everyone, I'm Julia. I'm a first year master's student under the Institute of Medical Science at U of T. My supervisor is Dr. Colin Hocko. So we're looking at individual variability in schizophrenia spectrum patients. So I look at functional neuroimaging data, and we're basically looking at how there's variability across functional brain activity within the spectrum of schizophrenia. So it's not a case control design, rather we're looking at the heterogeneity within the disease itself.
Hi, so I'm Robin. I am it's such a weird time. I'm between my master's and my PhD. I was a TA for HMB 300 2 years ago, and then I completed my master's and took a year to work in clinical research. I'm going back to the same lab for my PhD as fall, and I'm working with Dr. Eyal Grunbaum at Sick Kids. And my thesis is involving autologous gene therapy for kiddos with primary immune deficiencies. So where they would normally get a bone marrow transplant to correct their disease, we're trying to see if we can use their own stem cells transduced with a viral vector to be able to correct their disease. So lots of mass work here, but yeah, that's me.
My name is Fraser. I'm a fifth year PhD student in the Department of Molecular Genetics at U of T. I guess the professor I'm working for at the moment is James Ellis, also at Sick Kids. And what we do in our lab is kind of use induced pluripotent stem cells as a model system for studying autism spectrum disorders. So essentially what we would do is we get either blood or skin samples from patients who have ASD, as well as non-affected donors, so like their parents as kind of controls. And we can actually reprogram some of those cells back into stem cells. And then once you have the stem cells, differentiate those into cortical neurons and study how it is that the neurons that are derived from people with autism kind of grow and function and connect differently than those people who don't have autism. So my own project is kind of like looking into the mechanisms that are kind of driving how these neurons grow and behave differently, and try to study how they function in networks.
I'm Tyra. I am a second year PhD student in music and health science, but I'm part of the collaborative program in neuroscience. And my dissertation, I'm just working on figuring out my proposal more, but I'm looking into genetics, interactions between genetics and music training on learning motor skills and neuroplasticity. So some of my dissertation methods include using genome-wide array sequencing to look at genetic differences between musicians and non-musicians, and then also looking at the effect of short-term music training and long-term music training on brain-derived neurotrophic factor production. So, and also my PI is Dr. Michael Tout, and we're the Music and Health Science Research Center.
I guess we kind of like go around then and maybe in the same order, go through, what do you do on a daily basis? What's a typical day like for graduate students? And I guess if you work alone or if you work with others in your lab.
So I guess you guys, if you're in fourth year, you're probably going to relate to me if you're going to the same path. So I actually didn't get to spend a lot of time in my lab. Luckily, I'm a dry lab, so I can do a lot of stuff remotely. So I've been working from home from Monday to Friday, 9 to 5 kind of thing. My lab is a multi-PI lab. So generally, it's supposed to be a very collaborative lab. There's lots of RAs, there's lots of master's students, PhD students, postdocs. There is a lot of interaction. We have like weekly lab meetings, discussions outside of our normal lab meetings as well. So it's interactive, but we don't get to experience the full lab experience, for lack of a better word.
So when I decided this year that I was going to take a year to work, I also agreed that I would be keeping what would be my PhD thesis running in the background, which just creates a little bit of extra time. So it sounds like I'm in the lab a lot, and I am in the lab a lot. I'm here at Sick Kids 7 days a week between working and my thesis. I'm usually here between 9 a.m. and 6 p.m. and then I'll usually take a bit of a break.
I find in stem cell research, especially, when we're making cortical neurons from the iPS cells, like that's a very long process. A lot of our time points are like at 9 weeks post differentiation. So I find like with our type of research, there's a lot of kind of not having a ton to do for a few weeks, and then all of a sudden having a bunch of work to do when all your cells are ready. You need to process them. So like there might be some days where you come in for 4 or 5 hours, and you kind of go, well, I could stay here and read, or I could go home and read. And you might be okay leaving then. But then there are other days where it's like, you're there until midnight, 1 a.m. if you have like a lot of work to do, right? Our lab's fairly collaborative. There are a lot of times where, people kind of end up specializing in different techniques. And if someone needs like that technique done for their own research, they might just come to you and say, hey, you're able to kind of process these cells for me. And then, if you need their help, they can do that for you. So it's fairly collaborative. We also collaborate with a lab at the University of Western Ontario, who are more focused in like computational neuroscience. So we end up generating a bunch of data about like how neurons interact in networks. And then we kind of send that data over to them, and they're able to do some kind of interesting analyses with it, and then kind of come back to us with some interpretation.
Yeah, so my PI, like before COVID, really wanted us all in the lab as much as we can, but he was still really flexible. So I think most of us on average are there like 2 to 3 days a week. I think like our lab space is great for when we're collaborating. So my degree, the first 2 years is really exploratory. And then the last like 2 or 3 is where you're focusing on your dissertation stuff only. Right now, as I'm working on my own work, I am kind of in a solitary position. So in my program, it switches from being more collaborative earlier, and then there's a period of where I'm working on my own stuff. And then I think later on in my PhD, I'll have younger students help me and I'll be training them. So that's how my lab works. And my everyday, it changes as well. Like when I'm working on some more abstract tasks like writing, I find I can't be doing that all day. I'll get tired. So I'll break my day up into chunks. I have to manage my own time and say, okay, on Monday morning for 2 hours, this is going to be the time I work on this project. And on Monday afternoon, I'm going to work on a different project, or Tuesday is dedicated to X project. So that's how I manage my time.
I had a friend who flipped his sleep cycle and just come in at like, 11 at night and work until 7 in the morning. And like, that was fine. No one really cared, right? As long as you're getting your work done, that's totally okay.
John asked, what's the research coursework balance in grad school? I'm with the Institute of Medical Science, and for our master's program, we're required to have 2 semester full-on courses, and then 2 modular courses, which are like a 6-week course, like a mini course, and then it's credit, no credit. So we have 2 courses worth of credit, and then 2 courses that you get a grade for. And then once that's done, it's mainly your research. You don't have to do any more coursework.
I'm in immunology, and starting with their master's because they're phasing out their thesis masters and have more of a course-based master's to start. So when I started in that, it was very heavy into coursework to the point that there was probably 2 full days of coursework per week, and then the remaining days I'd be in the lab. So it was probably 6 courses in a year, or the 6 FCEs in a year. And I think even going into the PhD in immunology, they're quite heavy on their courses. You have a lot of the departments will give you electives, so it's still 2 full credits of electives. Plus, I think there are a lot of courses that you'll get graded on your attendance and your participation at seminars, a lot of research methods courses and statistical courses too. So quite heavy on the courses again. So I think I have another 5 full credits of courses as a part of my PhD. Yeah. So, but again, it's something that does change at different points. So it's one of those things where they're like, load up all your courses, maybe at the start of your degree. So later on, it's out of the way and you're not dealing with that on top of writing your thesis or anything else. So
in Mogen, they very much focus on like research first, and coursework is like kind of almost an afterthought. Where like we still have to take, I think it's basically 6, they call them like topic courses throughout your grad school if you're doing a PhD. So the way that most of my topic courses have run will be you'll have like a PI or two who's running the course. They send you out like a list of papers they want you to read in that week, and then you just show up to the seminar and you're expected to just discuss those papers, and just kind of talk about like what the kind of implications of them are. What does it mean for the field? Kind of like what you guys are doing, I guess, with you putting your critical review together, right? The evaluations for all those courses are mainly like, at the end of the course, you write like a one-page research proposal, and that's kind of it. Or you give like a presentation. So like I've never, in the 5 years of grad school I've never written a midterm. I've never written a final. Maybe we can move on to, what would you recommend for an undergrad student to do in order to prepare for grad school? One of the hardest things, I think, is getting used to being wrong a lot. Like, I feel like, with undergrad and courses that you normally take, like it's very much you do your studying, and if you've studied well and understood everything, you write your tests and like you hopefully get things right. With research, I feel like you can, have the best laid kind of plans and like a really good hypothesis about what you expect is going to happen with your research, and you do the experiment and it's like, oh, it didn't work, or I wasn't right about that. And so you have to be able to, I guess, not take that personally and kind of just understand that that's the way science works most of the time.
Advice would be, find a way that you can make yourself a routine day to day, finding a bit of discipline and be able to kind of lead your own work, because it only moves in, like, to a certain extent within your control, at the pace that you set it. So be able to kind of, take time for you when you need to, get up in the morning, stretch, eat breakfast, take care of yourself. And don't leave that to anybody else but you, because it's really on you to take care of yourself and to set everything for you at this point. So setting up those good habits early.
Okay, let's move on to the next question. We have a lot of questions. So the next one is, what type of research tools are used during COVID period online labs?
Yeah, the contingency plan for someone like me who's more of in a dry lab, like neuroimaging, human research, behavioral, stuff like that, was first, like right away when we had no access to lab at all. We were all kind of working on review papers or looking at database work. We've been able to have some behavioral research for a few studies that were considered priority continue, but it's still very much like looking at databases and trying to reanalyze old data, doing lit reviews, and writing my proposals. So I don't need to be collecting data right now, but there has been people who are in the year above me in my track have had their degree sort of delayed by a year, really, because they haven't been able to collect data, or they've had to transition their dissertation from being like a in-person study to an online study. So there's been a lot of development of new methods to get online behavioral research in psychology and neuroscience, done online telehealth studies. Last summer, I led a study that was a survey project asking music therapists and clinicians about their experiencing switching from in-person services to telehealth.
We've also had students who have had to make similar changes, like especially for, like, there were some grad students who I guess would probably be the same as Julia's experience, for like they had just started working in the lab and then all of a sudden everything shut down, right? And so they had originally planned to do all this like wet lab work, so wet lab being like actually doing experiments and stuff like that, and then all of a sudden they have no access to the lab and they're only doing a master's, and it's like, well, what do you do? So they ended up switching to doing more computational projects. So like we already have collected a bunch of gene expression data, sort of omics data from a lot of different cell lines that we have. And so their project ended up pivoting to, well, let's just analyze this data set that we already have, kind of like in a different way.
Move on to the next question. Do you think that research experience in undergrad helped you prepare for grad research? Would you recommend going into research-based grad programs if you have minimal wet research experience? I think that's the point where you determine, do I like working in a lab or do I not? I think that's really important, because you don't want to jump into a research-based grad program and then realize, oh wait, I don't even like doing research. I don't even like going to a lab every day. And in terms of minimal wet lab experience, I actually did, I started in my second year volunteering at Paul Franklin's lab, which is strictly wet lab. So I did wet lab mouse work for 3 years, I want to say. And then I actually went into a grad program that now I'm only doing dry lab research. So I don't think that if you have minimal experience in one aspect, that will necessarily impact you applying to different schools. I think you should get an equal experience, maybe doing both, so you determine whether you like doing wet lab work, whether you like doing dry lab work. Do you like working in a lab at all? Would you rather do something more professional based? I think that aspect of undergrad is really important, just to kind of gear you into a career.
I'll add to that and just say that I think, it's important to find out what you like, but I think undergrad research, it really gives you time to figure out what you don't like, which is just as important, because I like a lot of people that have gone into clinical research and been like, I really don't like this, or even going into wet lab and being like, I can't stand working with mice, which is totally fair. So just figuring out and taking that time to, I work with mice now, so I'm like, yep. To figure out if you really don't like something. I think if you don't have a lot of research experience, that's also okay. U of T has a lot of really great courses that are good in teaching research methods. So something like HMB 310, I think it is, teaches a lot of really great lab methods.
We can move on to what's the most valuable skill you've gotten or developed from grad school? That's a good one. I guess probably time management, even though that's something I'm still developing. But like, I think that's something that you definitely have to, have in check. Otherwise, things are going to be kind of rough. I also feel like just the ability to find information and digest information. I think it's something that's very translatable. Like, I know a lot of people after they finish their PhD will end up going into consulting, because I feel like, I could, if I went and did a postdoc in a completely different field, it would be challenging. But I feel like through the skills you develop in grad school, you'd be able to do the proper like literature sort of, deep dives into that field and get yourself up to speed with what's going on, and still be able to generate useful kind of ideas about different research questions that way.
Like I said, first year as a master's student, working strictly from home. My PI doesn't micromanage me at all, so I'm sort of on my own in the sense that if I want to sleep all day, I can. So I gotta like time management in a way that gets my project progressing.
I've been working on this year has been being better at asking questions. So if I need more clarification, but also in thinking critically, you go to a research seminar by someone who's doing really cool research, you want to be able to ask a good question to kind of figure out things. So I think that takes a lot of critical reasoning and a lot of critical thinking about the things that you're being presented, and also somehow how it fits into what you know, but also into the greater context.
I wanted to respond to like Eden's question here. She's asking about, do grad schools prefer if you have a single 2 to 3 year experience, or in one lab, or smaller experiences in many labs? I think when you're applying, the main thing is to be able to just set yourself apart from other people, right? If you have more than one experience, like how did you grow from one experience to the next? What skills did you learn in each of those experiences? And just being able to communicate what you gained from that. Speaking from me personally, like I spent 2 to 3 years, like so every summer, like volunteering as a Keenan research summer student. But I was sort of so low on the totem pole, I guess, that I didn't get involved in big projects, and I was still just doing some laborious tasks of database input like for those summers there. And then I think I felt like I had more experience from the single 1 year experience that I did as a work study student in a kinesiology lab, where I was running an experiment, analyzing the data. So I felt like there I did get a lot of the Keenan research summer student experience, like knowledge wise about neuroimaging, but I wasn't like, I didn't get a lot of, they didn't place me in a larger role where I could almost run a study. But in the 1 year experience that I did, I was able to like run a study for a grad student, analyze their data, put together something for them to present at a conference. So like I almost got more out of that short 1 year intensive experience than a 2 to 3 year like volunteer commitment.
I kind of had the opposite experience. I was in one lab for 2 or 3 years, and I was at the point where I was running the own labs in terms of behavioral testing. I was doing the perfusions, I was doing the cryostats, I was doing the immunochemistry. I was doing basically all of that, and then at the end, I'd just be like, "Oh, look what I found." So I was able to go through the whole experience, which is different from you. So I guess it also actually depends on what lab you're in as well.
Maybe we can jump to one of Professor Drew's questions just about like the application process for grad school. Yeah, so what was the application process like? I guess for me, I only applied to the one program because I got to the end of my undergrad and wasn't entirely sure about what I wanted to do. So I decided that, well, the application period for the U of T program was open kind of late. So I was like, okay, well, I can still apply to this one. And I ended up getting actually an admissions interview, which is kind of strange. Normally, it's just like you send in your application, you get a yes or no back. I think for me, because I had applied kind of late and my undergrad transcript wasn't spectacular, but I'd had a bunch of research experiences and a lot of really good like reference letters from the PIs that I'd worked with. They called me in for an interview just to see like, you know, what's up with that, right? So they asked me, you know, about my grades. They asked me about my research experiences. What did I find? And then they asked me if I had any samples of work that I had done, which thankfully, like I'd brought my undergrad research project thesis with me. And so I just handed it off to one of them and they were able to like read through it and be like, okay, this looks good. And I ended up getting an admission that way. As far as like what happens after that, so there's some grad programs where you need to find a PI that you want to work with before you kind of do the application. With Mogen, that's not the case. After you join the program, they basically set you up with a lab to do like a rotation in. So you work in a lab for like 4 weeks, and you do like a second rotation at a different lab for 4 weeks, and a third rotation. And at the end of it, you kind of do like a matchmaking process and you rank like your top professors you want to work with. The professors rank their top students that have worked with them, and they kind of, you know, match you up. So I was very lucky that I got my top lab of choice. It was kind of a nice process, I guess, because it allows you to, so like someone was saying before, not only see what you like, but what you don't like. So I got to work with a bunch of different model organisms. Like the first lab I worked in was a fly lab. The second one was C. elegans. And like I learned, I don't want to do C. elegans work. It was a lot of eye stress and just like picking worms and stuff. So yeah, it was nice being able to try out a bunch of different experiences before you commit to it for a long time.
IMS, you have to find your PI. So you get accepted, but your acceptance is always conditional on if you find a PI. So if you don't find a PI, then you can defer your acceptance for another year or whatever, but you have to have a PI before starting, and that's on you to find your own. And I believe physiology is the same. I applied to both physiology and IMS, and I believe both of them were the same. They were conditional on actually finding a PI.
Like you have some that are similar compared to like Fraser's program where you applied to the program, and others where you applied to the PI. And honestly, I think if you know like a general field that you're interested in, and a piece of advice I'd give you is to go to a conference. Right now they're online, which is great because it's accessible, but a little you lose out on the networking part. So the year before I was applying, I went to a conference in my field and met all these, pretty much like every lab I was interested in, like they were there. So I was able to meet with professors, have dinner with them, have lunch with them. And that was like more time I'd get with them to learn about their lab than I would in an interview almost. And it also helped me get my name in. They recognized my name and face when I applied. So that was useful. And that like helped me get an acceptance there, just like being able to meet people in person and talk to them about my interests. So just speaking from my personal experience, like networking and reaching out to these PIs before applying really helped my chances of getting in, especially when I didn't do well on the GRE. or if there's like a hole in your CV, maybe you have less research experience, etc. Just being able to convey your skills so that when you send something in on paper, it's not put to the side.
Okay, we have one more question. So does your research result in a lot of collaboration with other departments or universities, or is it pretty self-contained? Do you find that there's a strong collaborative sentiment in your research area, or is it more competitive?
I found that at least in Toronto, it's very, very collaborative between different labs that are working around here. I've never felt like I couldn't go and talk to members of a different lab about my own research project out of fear of getting scooped on whatever it is I'm working on. I don't know if other people have different experiences with that. I feel like in the United States, it's a lot more competitive. I've heard some horror stories from people working in labs there about professor will hire 2 postdocs to work on the same project and will just say, all right, whoever gets the results first gets to be on the paper, and there's competition within labs. But I haven't heard anything horrible like that in Toronto. I feel like everyone here is very open to working with other labs just to get the best results that you can.
I think that's a big shift from the undergrad scene at U of T also, is shifting from, competition to collaboration, and all your peers, and bettering them is also bettering you. So definitely collaborative. I find also I have a lot of opportunities to collaborate within SICKIDS, but also internationally. These are very well-ranked institutions throughout U of T as a part of their grad schools and the hospitals that are affiliated. So something like part of my project, we have a collaborator at UCLA who's letting me come study there for half the year next year. So you have a lot of really cool opportunities to go meet a lot of really cool people doing amazing work. And U of T is a very good place to be able to, put yourself within that network.
We're very collaborative, but those collaborations have to be facilitated just to make sure that our lab still has control.
We have one final question. I guess before it hits 11 o'clock, what does your department or lab normally expect students to produce in terms of things like publications, presentations at conferences, etc., before finishing their master's or PhD?
Yeah, for Mogen, I think the requirements are fairly explicit. I think for a master's, they want you to be able to produce one publishable figure out of whatever work you do in your master's. For the PhD, usually they need you to have some kind of first author publication in a fairly reputable journal. I know I've heard the unofficial guidelines is basically the number of first author publications plus your number of years in the program has to equal 7, which isn't a hard thing, but it's if you have one first author publication out of 6 years, I think the average completion time for our program is 6.5. Then that's good. But if you're extremely productive and are able to pump out a bunch of publications, then maybe you don't have to spend quite as long in the program. But yeah, you basically need to have enough data collected to be able to defend in your thesis.
I think in terms of conferences, IMS requires their master's students to present at least once.
I think our dissertation's supposed to be able to get us 3 publications. Whether that's one of them is the lit review chapter of our thesis. And they don't all have to be experimental, but we should have 3 publications from our dissertation. But the goal is to have 3 first author publications from our thesis.
I think immunology is the same too, so 3. And then conferences kind of come, I think, later as you get into your thesis, will be more common and everything. So
I guess it's 11 o'clock. Do we want to try to address the one last question Professor Drew had, just as we're kind of on that topic anyway, about we keep hearing how graduate students are supposed to publish papers. How hard is that, and what's the difference between that and a thesis? It really depends on your field as well, about how I guess what the expectations are, and maybe how long it takes to put those papers together. So I have friends who work in labs that do brain organoids, where many of your time points or experiments are okay, we need to wait for these organoids to grow for 6 months before we can slice them and stain them. So putting all your, like, getting your replicates together, do your experiments, putting all this stuff together, it takes a very, very long time just because of how long, these experiments take to put together. A lot of mouse work too. It's like if you have to wait for mice to grow and develop and breed to do your research, I mean, it can take a long time. I know, depending on which journals you're actually submitting to as well, some of them have very, very intense review processes where, like, we published one paper where I think it was in review for 2 years, just kind of going back and forth between people, saying you need to do more experiments. We do the experiments, get sent back to say, yeah, this is okay, but maybe you want to publish it actually in this other journal instead, which means it has to go through that process again. And yeah, it can take a very long time.
To clarify, like, the difference between your thesis and publications is, I think this thesis can have, for your master's, like, typically it's like one study and you're trying to using the study to prove a theory, and the thesis will be like up to 100 pages in length, 50 to 100 pages of length depending on your department, and it'll be more in depth versus a paper that you might see in a publication that might only be 10, 20 pages. So it's more of a concise version, I think. And then also for the dissertation, this varies by department as well, but your dissertation is a collection of different studies where each of those studies can be a publication. So I just got my master's thesis essentially accepted like last week, and that was like 10 pages, but my master's thesis was like 60 pages. So it actually wasn't that hard for me to take my master's thesis and write it up for publication. And also your thesis is adjudicated by your committee, so your supervisor and other professors at your university and external to your university, versus peer publications are peer reviewed by a journal. So that's also a difference there.
I think your involvement in publications can be variable, where your thesis is completely, it's your own work.
I've also heard people doing the opposite too with their thesis and publications, where, like, for your PhD thesis, you typically will have 2 data chapters that you have to have. And so, like, if you've published 2 publications where you're fairly high up on the author list, you can kind of like just not copy and paste the whole paper and put it in there, but essentially just rewrite that paper that you've already published as part of your first data chapter, second data chapter.
I think we're going to wrap it up. Thank you guys for all of your questions. This was really fun for all of us. Does anyone have any last words before I stop recording?
No.
We're all good?
Yeah,
Okay. Thank you guys.
Thanks.
See ya.
Bye.